I'm Steve Brown, Technical Editor of Spectroscopy Magazine, and I'm here today with Emily Smith, Assistant Professor in the Department of Chemistry at Iowa State University. Emily, thank you for joining us today.
Thank you.
Your presentation discussed a technique called stimulated emission depletion fluorescence microscopy, which you use to collect subdiffraction fluorescence lifetime images. Can you briefly describe the technique?
Sure. The main idea behind stimulated emission depletion imaging is to use two laser pulses. One of those pulses turns the fluorophore on, and the other pulse turns the fluorophore off. The key to STED is the way those two pulses are shaped. So the pulse that turns the fluorophore on has a Gaussian profile, and the pulse that turns the fluorophore off has a donut profile with zero intensity in its center. So when you overlap those two profiles, what happens is you get stimulated emission, or you turn the fluorophore off at the periphery of the excitation profile. So then you're collecting just the spontaneous fluorescence at the very center of the excitation profile, and those dimensions can be smaller than the diffraction limit of light. Spatial resolution in the tens of nanometers is possible. We've reported 40 nanometer spatial resolution with our imaging setup. So the main idea behind what my collaborator, Jake Petrich, also in the chemistry department at Iowa State University, what we've done is we've binned the spontaneous fluorescence signal that we're collecting in time. And what that allows us to do is extract the fluorescence lifetime in subdiffraction volumes. So we're able to do subdiffraction fluorescence lifetime imaging with 40 nanometer spatial resolution.
Great. How does the technique compare with traditional confocal microscopy in terms of resolution and other parameters?
So the closest imaging techniques to what we do is fluorescence lifetime imaging, or sometimes called FLIM. And that's a diffraction-limited imaging technique. So the spatial resolution is going to be somewhere in a couple hundred nanometers, let's just say 200 nanometer region. So you're not able to distinguish between two objects or two features of an image if they're separated by less than 200 nanometers if you have a densely population of fluorophores. So instead, lifetime imaging, which is the technique that we do by overlapping our two pulses, we can get spatial resolution in the 40 nanometer range. So now we're able to distinguish between two objects that are separated by 40 nanometers or up to the diffraction limit. So FLIM is a very commonly used imaging technique. It's used by a lot of biologists, a lot of material scientists, and just diverse sciences use fluorescence lifetime imaging, but in the diffraction limited version. So you can envision that all kinds of different systems or different types of experimental problems could be studied if you're interested in getting spatial resolution below the 40 nanometer spatial scale. So other than the difference in the spatial resolution between FLIM and STED lifetime imaging, the parameters are pretty much similar. And so again, this opens up the possibility of all kinds of different systems, different biological samples, different types of samples that might be studied by lifetime imaging.
What are the main challenges in performing this type of microscopy?
So the technique referred to as STED, or stimulated emission depletion imaging, was first pioneered by Stefan Hell many years ago. And there are challenges to doing STED imaging, and then I'll maybe address the challenges to doing STED lifetime imaging. So in STED imaging, the shape of your two pulses, the one that turns the fluorophores on and the one that turns the fluorophores off, is very critical. So you have to have a STED profile that has a true zero at the center so that you don't decrease your overall signal, but at the same time you do get improved spatial resolution. It's also important that you have a certain temporal profile between those two pulses. Related to STED and STED lifetime imaging, we have to gate the signal so that we are getting only the signal from the fluorophore. And at the same time, we have to collect enough photons so that we can extract a meaningful lifetime from each of the pixels in our image. So the challenge to getting enough signal is that we actually are hitting the molecules with a lot of photons, both the excitation photons and the STED photons. And this can lead to photobleaching. So you have to use pretty stable fluorophores and trade off between the amount of time you're collecting the signal and the amount of time that you're exposing the molecules to the laser, because that will cause photobleaching. So we were able to find all the conditions, the balance, the trade-off between those two criteria that we were able to extract meaningful lifetime from fluorescence lifetime images.
What types of samples have you analyzed, and what sample types are most suited for this approach?
So we have only analyzed cultured cells so far, cells that have been grown in cell culture medium, that have then been fixed or killed, and then stained so that their actin cytoskeleton, the protein structure within the cell, has been labeled with a molecule called phalloidin, and the phalloidin then is bound to our fluorophore. So we've used dead lifetime imaging to determine the structure and organization of the actin cytoskeleton at these subdiffraction spatial scales. Another interest area of my research group is measuring the organization of the cell membrane. And one of our long-term goals is to make a connection between the organization of the cell membrane, the proteins, the lipids, the small molecules in that organelle, and the organization of the actin cytoskeleton. So far we're just at the beginning steps of making that connection between the organization of the cell membrane and then what goes on inside of the cell. Samples that are amenable to fluorescence lifetime imaging, as I said before, would be perfectly suitable for our STED lifetime imaging. So there's really quite a few different types of samples and different research areas where this technique could be applied.
What are the next steps in your research with this technique?
One of the challenges I mentioned to STED is getting enough signal so that we can extract meaningful lifetime from our STED lifetime images. And one of the things that we've recently demonstrated is that we can use quantum dots. These are inorganic nanoparticles. They're commonly used as tags in fluorescence microscopy and fluorescence experiments. They're very stable, so they don't tend to photobleach, even after long exposure times, even high laser powers. So in collaboration with another chemistry professor here at Iowa State University, Professor Javier Vela, we have shown that quantum dots, they're called giant nanocrystal quantum dots, can be used in a STED and STED lifetime imaging format. And we were able to get 40 nanometer spatial resolution with these quantum dots. So what that allows us to do is collect a signal for fairly long periods of time, so we get very high quality fluorescence decay curves, and we can then extract fluorescence lifetimes and even complicated, bi-exponential or complicated lifetimes from those curves. So we've demonstrated that we can do that, and our current work is trying to understand the mechanism behind that improved spatial resolution. The photophysics of these quantum dots are a little bit complicated, and so we now want to understand in a detailed way what is the mechanism for getting that improved spatial resolution. So we know that we can turn the fluorescence off at the periphery of our excitation profile, but we don't know the mechanism for why that's occurring. So we're trying to determine what that mechanism is, and then that could open up the possibility of using all kinds of nanoparticles that are fluorescent, other types of quantum dots and other novel nanoparticles in the STED format. And so that should be pretty novel, and we're pretty excited about the ability to collect this high-quality data.
Very interesting. Emily, thank you for speaking with us today.
Thank you, Peter.